So actually most of the groups here are the glyco groups, so we know it. So this is mostly for our guests and for our students to learn something new. The idea is to present our current analytical capabilities and some examples how we can do something useful for our clinical colleagues to develop some kind of a collaborative studies. I will deliver something like an introductory lecture to the glikobiology as a field, including some of the examples from our recent work in our lab. So I will start with a typical picture I use to show how things are wrong. So this is something you can find in a textbook. And people believe in this picture, and we know it is wrong. What do we know that it is wrong? We know that these tiny little things, which are shown here, like an addition, do not look like this. Glycans are not tiny little modification on a cell membrane. When you look into the real picture, so when you look into the microscope, electron microscope photography of a membrane, this here white tiny thing, this is a cell membrane. And the big dark layer on top of it, these are the glycans. So glycans are not tiny insignificant things on the surface of a cell. Glycans are covering all the cells, so the picture should look more like this. You have a membrane, you have some proteins, and you have a lot of different glycans on top of it. Glycolipids, glycoproteins, protoglycans, everything there is, it's mostly glycan. So, as a minimal baseline, we should all agree to that the glycans are one of the 4 principal components of the cell. You have DNA, you have proteins, you have lipids, and you have glycans. And practically anything on the surface of a cell has to deal with glycans. So if another cell is approaching the cell, it will communicate with glycans. If pathogens are approaching the cell, they will bind to glycans. All the hormones, all the signaling molecules, they have to deal with the glycans on the cell membrane before they can actually enter the cell. So the question is, why are glycans being ignored by the majority of the mainstream science? And the answer is not so easy, but I think the simplest answer is because glycans are so complex. They are structurally extremely complex. Normally, proteins, DNA, these are linear structures. You write down the letters and you know the structure. When you think about glycans, you have to think about the branching, the number of branches, the position of branching, the type of linkage, the anomeric configuration of a linkage. There is a huge information capacity in each glycan molecule. And at least 2,000 different glycan blocks can be found on a protein. So compared to 4 nucleotides, 20 amino acids, you have something like 2,000 glycan blocks which are combined in different ways on the glycoproteins, and you get a glycoprotein which is several orders of magnitude more complex than the protein itself. So this is something which is at the moment beyond our comprehension. We cannot analyze it, we cannot understand it in a full detail. Even the single glycoprotein, simple IgG, with one conserved N-glycosylation site on the Fc region, can theoretically be produced in 900 different glycophores. So 30 structures, 2 sites, 900 theoretical glycophores.